Yes, in the interest of time, would help if you could see the screen. In the interest of time, I am going to try to move through a few things kind of quickly. And if I get to the end of what I want to cover before our actual end of time today, I might go back and add a few things in, but we'll see. So to remind myself, we finish at 4:15, right? I want to keep that in mind. All right, so female reproductive system. This is a sagittal view, and it's kind of a weird little picture, what they're trying to do. In the sagittal view, you would not see the ovary really, because it's not on midline, but they're putting it there. And then I'll show you the other stuff that has to do with it. But on midline, what we do see is the uterus, which has a very thick muscular wall, which opens up at the cervix into the vagina, which is another muscular walled structure, which is essentially the birth canal. This view, anterior view, shows everything too. And the ovaries are here on either side in that kind of bluish color that I pointed out a second ago. These are the reproductive glands of the female system. They're where the eggs are made, where meiotic divisions lead to the eggs. And we'll talk about that process in a little bit more detail. They're analogous to the testes in that they're the gland where meiosis takes place. But they're different from the testes because the testis, like I said earlier, is really just a bunch of tubes inside the membrane. The ovary is more of a glandular structure that the egg cells emerge from. It's almost like, and I don't think this is entirely fair representation, but it's almost like the testis is an exocrine gland because what it's producing and secreting is eventually going to leave the body through the reproductive tract. And the ovary is an endocrine gland because what it's producing and secreting stays within the system, okay, up to a point. But really when we talk about endocrine glands, we're talking about hormones, and there are hormones that are released from the ovary. It also produces and releases eggs. So it's not entirely correct to say one's exocrine, the other's endocrine, but they kind of do fit that a little bit just in the structure of the gland. One has tubes or ducts like we'd see in an exocrine gland, and the other one's more sort of a solid glandular structure like we'd see in the endocrine system. Now, when the egg cell emerges from the ovary, it moves into this tube here, which is labeled as the uterine tube because it connects to the uterus. That's also known as the fallopian tube. And then in parentheses there, it says oviduct because it's the duct that the ovum or the egg cell moves along. The uterine tube isn't connected directly to the ovary. There's a little bit of space between them, and it's possible for the egg cell, when it's released, to not make it into that tube, and it'll move into the pelvic region. If sperm get all the way through the system and into the pelvic region also and end up fertilizing that egg cell, that can lead to an ectopic pregnancy, which is a bad situation. But most eggs that are released from the ovary and that don't get into the uterine tube will be reabsorbed and they won't cause any problems. The end of the uterine tube has these structures called fimbriae, which are kind of like fingers that help to coax the egg cell into the tube, and then it'll migrate along here. Fertilization actually takes place in the tube, and then the fertilized egg cell moves along and eventually gets into the uterus. The picture I showed at the beginning that had the 2, 4, 6, etc. cells, the first row were divisions that take place in the uterine tube, and the second row were divisions that take place in the uterus. Just those are sort of time points where things are happening. And then here we have the uterus, and it opens up at the cervix, which goes into the birth canal or vagina there. The book goes on to talk about the external structures. I'm not going to deal with that. I normally don't. I just want to concentrate on the internal structures. But after this picture in the book, basically it goes on to show the structure of the labia and shows the extent of the clitoris, which is analogous to the erectile tissue in the penis. The clitoris is made out of the same kind of tissue. From the picture in the book, you can see it's actually quite extensive and wraps around a lot of the other structures. This picture is not in the book. I got it from Wikipedia. It's sort of a summary figure of what happens in the uterus in producing the egg cell. And I'm not really crazy about these kind of pictures, but I think they are kind of helpful to show you everything that's happening. What's depicted here doesn't all happen at once and does not move through the ovarian cortex like this picture suggests. Really what they're showing is the first step, the second step, the third step. They're just drawing them all in one picture, but it's progressing over time. In the cortex of the ovary are egg cells that were produced in the embryonic ovary that are waiting to be activated to go into this the ovarian cycle from month to month after puberty. As they're sitting and waiting for that to happen, they're contained within what's called a primary follicle, and then hormones with the recurring ovarian cycle will stimulate those not primary follicles. They're called primordial follicles, which is not in this picture. But when they're stimulated by hormones, they'll start to develop and they become the primary follicle, which grows and becomes more complex. We'll look at some micrographs in a second of what they look like. Eventually, the follicle matures to this full size where it's right up against the edge of the uterus, ovary, and then at the halfway point of the cycle, so generally we think of the cycle as taking 28 days, so on day 14, the mature follicle ruptures and releases the ovum or the egg cell from the ovary, and it'll then move into the uterine tubes, like I was saying a second ago. The rest of the follicle, what's left behind after ovulation, persists as this structure called the corpus luteum. Corpus means body or structure, so it's this thing. And then luteum refers to the fact that it has a yellowish color. So it's the yellow body is basically what that means. But it's releasing hormones from the midpoint of the cycle through the rest of the cycle to help regulate what's happening for the second 2 weeks, the third and fourth week of the cycle. Hormones being released from the follicle before ovulation, and between the follicular and luteal hormones, that regulates what's happening in the uterus, which is either preparing for or going to support the ovum when it implants there until the embryo starts developing. Now, one reason I don't like these kind of pictures is because they make it look like things are moving through the cortical space. Actually, where the primary follicle is, if that's where the thing started, that follicle would grow larger in place. It wouldn't migrate along. They just did it that way to fit it all into the picture. Sort of in a similar fashion, with the way that the book depicts this, shows sort of a progression through the follicles, but it's not showing it to you in the ovary. So this is a little bit better representation of it. And so here we see the primordial follicle, which is the oocyte, which is the Greek word for egg cell. Early on, before it's going to start continuing to develop, the oocyte that we see here was formed in the embryo before that embryo became a newborn daughter and then grew up to be a woman. The cells are already in place there. And then with puberty, the primordial follicles are stimulated to go through development with each ovarian cycle, okay, which we'll talk about a little bit more. Surrounding the oocyte in the primordial follicle are a few cells which are labeled granulosa cells here because that's what they're going to become. But granulosa refers to the fact that they look like granules or grains. And they aren't going to look like that until the follicle starts developing. So these round cells here that kind of look like a grainy ring around the cell, those are the granulosa cells. This is what they look like before the follicle starts to develop, but that's what we see. The follicle will continue to develop and become, so it starts off as a primary follicle, and then it becomes a secondary follicle. And at this point, we know it's a secondary follicle because first off, the granulosa cells have divided enough to be several layers of cells, and then they start to separate from each other, and a space opens up within the follicle. Somewhat unfortunately in this picture, they make it look like the oocyte's floating in the middle of the follicle, but in fact it's not. It's attached to the side. It just happens to be we're not looking at a section through this where we can see it attached to the side. And as that secondary follicle develops further, it becomes the tertiary or mature follicle, which is the one I pointed out a second ago, that's right next to the edge of the ovary that's about to rupture. So tertiary refers to it's the third follicular stage, and mature is referring to the fact that it's about to rupture. And in this picture, we can actually see how the oocyte is connected to the edge of the follicle through some granulosa cells, but it's surrounded by that empty space. In this picture, as opposed to the one of the secondary follicle up above, you can see that there is some stuff in that center, and it's cellular debris and fluid that's accumulated in that space. But there is some space that's grown in the granulosa cells region. Now, in the previous picture I was just showing you up here, and in the one I'm pointing at now, they're pointing to some cells that are right on the edge of the follicle called theca cells, or some sources will call them thecal cells. And these cells help with some of the hormonal chemistry that's taking place in the follicles. Then the tertiary or mature follicle will rupture, and that's ovulation. Now, these other pictures that I've been showing you here are basically micrographs that were traced to show you what things look like. And we're looking now at a micrograph of ovulation. However, it looks very different because you can't really get the same kind of image of a rupturing follicle as you can of a developing follicle. The tissue that we see in the other pictures was prepared in the traditional way that the tissue is harvested and sectioned and stained and put on a slide and all that sort of thing. To see ovulation in progress, you have to actually take a picture of living tissue. If you're preparing the tissue and you're working with dead tissue on a microscope slide, you're not going to see ovulation. You have to actually see it in live tissue. So that's a light micrograph of ovulation taking place sort of in 3 dimensions, and it looks kind of strange. The dark spot in the middle, it's labeled oocyte, is in fact the oocyte, and it's surrounded by granulosa cells, which is just depicted here as a sort of cloud around it. But some of the cells leave the follicle with the oocyte there, and others remain behind as the wall of the ruptured follicle. And those remaining cells end up becoming the corpus luteum. Okay. And in this picture, that's all of the parts that they show you. The first picture of the primordial follicle is before the cycle starts, and then the primary, secondary, tertiary / mature follicles, and ovulation. That's all that happens in the first 14 days. And then the second 14 days is when the corpus luteum is there. It doesn't change that much, so they don't really have a bunch of pictures of it. It does, towards the end of the full 28-day cycle, end up kind of degenerating away and becoming a smaller sort of scar-like remnant of what it was. The corpus luteum will persist longer if implantation takes place and the uterus is actually going to gestate. The corpus luteum will stick around for a couple of months at least, helping support early stages in pregnancy. But if fertilization doesn't take place, then it degenerates away at the end of the 28-day cycle. The micrograph here is of a secondary follicle, and we know that because the antrum, or the space within the granulosa cells, is starting to separate out. There's less space in this picture than in the picture that they suggest up here, just taken at a different time. And so there's more obviously connections between the oocyte and the rest of the follicle there. But that's what we see. I just want to go back to this picture here. This developing follicle would represent a secondary follicle, and we can see the oocyte that's actually sort of in a little bulge in the wall of it. Like I said, it's just not represented in the picture from the book. And that's just a matter of where you happen to section through the tissue, whether you actually see the place where it's connected or not. Okay. Oh, I skipped over this picture for some reason. This is a representation very much like the one for making sperm, but of how egg cells are made. So again, we have a stem cell, and it's called the oogonium instead of the spermatogonium, because oo is the prefix that means egg in Greek. And it separates into, or divides into, two cells through mitosis. The labeling here is a little funny. It says primary oocyte next to the cell that has the arrow pointing back to the oogonium. That's not actually the primary oocyte. That's the stem cell that persists. The other cell is the primary oocyte. But they don't have a label on the other side of that cell because that stoplight with meiosis 1 begins there. So they just put off the other side to make it fit. But the cell that progresses through the rest of this at that stage, that's the primary oocyte, not the one that returns back to be the stem cell. So at that point, the primary oocyte starts meiosis. The first thing that happens meiosis is, well, first the DNA duplicates, and then the chromosomes condense, and they segregate out pairwise. And then the pairs of chromosomes, the maternal and paternal chromosomes that are matching up, can actually swap a little bit of DNA. So remember the blue-red exchange taking place in a few of them in the picture I showed you earlier? That's happening. That's what's going on during prophase 1. All of that's happening before birth. That's happening in the embryonic tissue before the daughter is born. Then meiosis stops at right there, right after it began in prophase 1. And it does nothing until after puberty. Now, that line there that says before birth on one side and after puberty on the other represents the girl's childhood. And nothing's happening in the ovaries. The cells are just sitting idle, waiting for puberty to start. Now, when puberty starts, not all of them start to do something. Then they're recruited month after month to go on through the rest of this process. Some of the cells that arrest in prophase 1, right before birth, sit there through most of the woman's life, all the way up till her last ovarian cycle before menopause. And they're just sitting there doing nothing until they're stimulated by the hormones. And then when they are finally stimulated, whenever it is, whether it's her first cycle or her last cycle, they move on to finish meiosis 1 and divide into 2 cells. Now, in the sperm picture we were looking at before, they designated the haploid cells with one N, and here they just designated as N. Mathematically, you multiply something by 1, it's the same thing. So N, 1N mean the same thing, which in humans, again, is 23. So yeah, a little discontinuity between the two pictures, but it doesn't mean anything different. After the first division, the cells we have now are referred to as the secondary oocyte and the first polar body. They are not identical cells. Production of egg cells is an asymmetrical process. The first polar body is really just the other half of the DNA. So the diploid cell that starts meiosis 1 divides into 2 haploid cells, which just have half of the chromosomes from the original cell in each. The first polar body is just half of those chromosomes that we don't really need anymore, or I don't need, I'm not a woman, but are not necessary for anything else in the reproductive system. They might go on to divide again, but not necessarily. The secondary oocyte is the other half of the chromosomes, and really all of the cytoplasm of that primary oocyte. Okay. The asymmetric divisions that we see here is to be sure that the egg cell has all the cytoplasm it can. Okay, remember the picture I showed at the very beginning with a really big egg cell surrounded by a bunch of sperm? It's really big because it becomes huge, keeping all of the cytoplasm through all the divisions we see here. So the secondary oocyte is then going to be what gets released at ovulation. Okay? It hasn't started the second round of divisions yet. Okay? It has to be contacted by the sperm. Fertilization has to start for the second meiotic division to even take place. If that egg is released at ovulation and fertilization never occurs, it never finishes meiosis. So the secondary oocyte is what comes out, and it's passed on with the menstrual flow or resorbed into the tissue or something like that. When the egg is penetrated by the sperm actually stimulates the process of the second meiotic division. And so that newly divided nucleus is going to combine with the nucleus that the sperm cells bring along. There's another polar body that gets produced there, which is the other nucleus from that second meiotic division, which again can kind of be a throwaway thing. And all of the cytoplasm stays in what's now the mature ovum. And all that cytoplasm is necessary to support growth of the early zygote. We call it a zygote at the beginning until it becomes an embryo, and then the embryo becomes a fetus, and then the fetus becomes a newborn. The fertilized egg cell, as it starts dividing, is a zygote until it reaches the embryo stage. And it needs all that cytoplasm because when it divides the first time into 2 cells, just splits that cytoplasm between the 2 cells. And then those cells divide and it just splits the cytoplasm. The cells don't actually grow much bigger than they were originally. A lot of the growth takes place later, but these cells, they keep dividing into smaller and smaller bits, but they're not going to just disappear because it's, to begin with, it's a huge cell with all that cytoplasm. So on the other side of the box where the second polar bodies are, those may or may not be produced, and it doesn't really matter. As far as I know, there's no real function of the polar bodies, but we might discover later on that they do have a role to play. And if you look back at the pictures I showed you of the divisions with the 2, 4, 8 cells, et cetera, in the picture with the 2 cells that divided from the fertilized egg cell, there are 2 other little cells next to it. So there's actually 4 cells there. The 2 that are the division of the fertilized egg cell, but then there's the 2 polar bodies. which I believe is the first polar body still hanging around, which never divides, and the polar body that's produced in the second meiotic division. I think that's what they are. They're there, and it's kind of unusual for things to be present in a situation like that and not play some kind of role. So I wouldn't be surprised if sometime in the future we discover that polar bodies are very important for some aspect of embryogenesis or something like that. But I don't know of anything specifically just yet. So I actually kind of meant to talk about that before I got into the follicle development stage, but doesn't matter. It kind of puts it all there. I do want to mention a couple of things. I already did sort of. All of the cells that are present in the ovary that are going to go through ovarian cycle, meiotic divisions, are present before birth, okay? So a woman giving birth to her daughter is also giving birth to all of the egg cells that could potentially be her grandchildren. They're already present in the ovary of her daughter when she's giving birth to her. That blows your mind so far. And then the other thing I mentioned, the second meiotic division only occurs when the sperm cell's there to fertilize it. If fertilization doesn't take place, then it never completes meiosis. These pictures show the relationship of the glands and hormones that regulate these things. And I showed you the picture briefly at the end of the male system that showed the hypothalamus and pituitary gland releasing hormones influencing sperm production. The same sort of thing's going on here. We just don't have an anatomical picture. But the hypothalamus is releasing that same GnRH, which is stimulating the anterior pituitary to release FSH and LH. FSH and LH, and mainly FSH, stimulate the follicles to go into that developmental process. And in fact, FSH stands for follicle stimulating hormone, okay, because they stimulate the follicles to do their thing. LH is present and plays a minor role here, but it's really more important later on. Bless you. When the follicles are developing, they start producing estradiol, which is a specific estrogen hormone. Estrogen is not a hormone. Estrogen is a family of hormones, and there are a few different ones. The main one that's produced by the follicles from the ovaries is estradiol. What estradiol mainly does is it then targets the uterus and affects what's going on there, which I'll talk about in a little bit. But also estradiol feeds back to the pituitary and the hypothalamus, inhibiting the release of these hormones, just acting as a negative feedback loop. Estradiol levels continually go up. So when the follicles first start developing, they start secreting a little bit of estradiol, but as they progress through to the secondary and tertiary / mature stage, the estradiol levels start to get really high. So early on, the first, I don't know, maybe 10 days, estradiol levels are growing, and they act as an inhibitory feedback signal. Then when they get to their peak level of expression, estradiol will actually activate the thalamus and sort of turn it back on. When they're inhibiting, they're causing GnRH and FSH and LH to not be released. They're not telling them to not be produced. They just get stored in those glands to be released later. And it's with the high levels of estradiol that come along later on that stimulates the release of the GnRH, which also in turn stimulates the release of LH and FSH. Now in this picture, the arrow for LH is a little bit thicker than the arrow for FSH. And that's on purpose. The amount of LH that's stored and released with this burst of estradiol signal is greater than what FSH does. So really LH is responsible for the next thing, which is it tells the follicle to ovulate or to rupture. So LH is really the hormone that causes ovulation. Okay. The follicles are still releasing estradiol that are still affecting, and that's still affecting the uterus also. Okay. After ovulation, in the ovaries, it's no longer a follicle. At this point, it's now a corpus luteum, and LH stands for luteinizing hormone because it's LH that caused the formation of the corpus luteum. Okay. It caused the formation of the corpus luteum by causing ovulation, and what's left over after ovulation is the corpus luteum. But that's where the name comes from. The corpus luteum then starts to secrete progesterone, which is a different hormone. It acts as a negative feedback signal to the hypothalamus and pituitary, slowing down the release of the gonadotropin release above. It also has an effect on the uterus to cause changes there. And I want to talk about the uterus in a little more detail. But basically, the first half of the ovarian cycle, estradiol is the main hormone that's being released. And after ovulation, progesterone is the main hormone being released. When we're talking about the ovaries, the first half of the cycle is called the follicular phase because that's when the follicles are developing. And then after ovulation, the second half is called the luteal phase because that's when the corpus luteum is present. We're going to talk a bit more about hormones, and we're going to look at graphs showing hormone levels over the 28-day cycle. So hopefully, if you're a little confused here, you'll see it explained in a different way in a little bit. But before I go there, I want to talk about what's going on in the uterus. Okay. In this picture, they have a micrograph inset of the ovary showing ovarian cortex tissue, which we're past that now. What's more important here that I want to talk about is this inset of part of the wall of the uterus showing some of the structure over here, and also what the tissue layers of the uterus are called. So we've looked at a lot of different organs in this semester and talked about the tissues that make up the walls of those organs. So blood vessels, digestive system, bladder, et cetera, et cetera. The layers of tissue that make up the uterus are the endometrium, the myometrium, and the perimetrium. Metrium in all three of these words essentially means uterus. So the endometrium is the inner lining of the uterus. The myometrium is the muscular layer of the uterus. And then the perimetrium is the outer covering around the uterus, which is essentially kind of like a serous membrane around it. So the perimetrium is like a serous membrane that we've seen in other organs. The myometrium is like the muscularis in the digestive system, or the myocardium of the heart, or the tunica media of blood vessels. The endometrium is a mucous membrane with lots of connective tissue. Unfortunately, in this micrograph, we can't quite see too much detail in the myometrium, but you can kind of see there's sort of a line here separating a darker from a lighter area. The darker part here is the myometrium. That's muscle tissue. And so all of the lighter stuff out here is the endometrium. And if we could see more detail of this, it has a simple columnar epithelium lining its outside surface. That's the mucosal lining, the mucosal epithelium of the uterus. And then all of this is connective tissue, which is actually segregated into two different regions. The deeper region, which is adjacent to the myometrium, is called the basal layer, or some sources will call it the basalis, which is just a Latinized form of basal. That layer doesn't really do much. It's sort of the base of the endometrium. It doesn't change. The rest of it from the basal layer out, which might be about this much of the tissue, is called the functional layer, or the Latinized version of that is functionalis. The functionalis, I should stick with the functional layer, the English version of that word, is the part that changes throughout the uterine cycle. Okay. Now, we're talking about the ovarian cycle, which was the follicles and the corpus luteum and all that stuff, which happens over 28 days. The uterine cycle is just the 28-day cycle that's tied to the ovarian cycle and to the hormones I was just talking about. And the hormones that are released from the ovaries, so the primarily estradiol in the first half and primarily progesterone in the second half, have an effect on the endometrium. And what they do is they regulate its growth and removal. At the end of a cycle, the hormone signals shut off, which stimulates the functional layer of the endometrium to detach from the inside of the uterus. And so the beginning of the uterine cycle is the menstrual flow, it's menses, 5 days or so, roughly. And then after that time, somewhere in the 6th or 7th day, the endometrium starts to grow back in width. And we call that the proliferative phase. So that's what estradiol is doing. It's stimulating the endometrium to regrow. Okay. During menses, estradiol levels are pretty low anyway, so they're not going to cause much growth. But once the estradiol levels get high, that'll cause growth. After ovulation, there's still estradiol present in the system and still being released from the ovaries, but it's minor compared to the progesterone that's being released. And what progesterone does is it stimulates the endometrium to start becoming a secretory mucosa. So there are glands in here, which you can kind of see. This is probably the neck of a gland. This is the luminative part of a gland. And there's, we could see more closely, we'd see a lot of little mucosal glands down in there. And so they start to produce secretions stimulated by progesterone, which prepare the endometrium for the possibility of implantation of a zygote and then gestation of the embryo and fetus. That will contain nutrient molecules to help the zygote and embryo survive until a connection's made across the placenta with the mother's circulatory system, as well as other things to help with implantation and survival. And that's what the second half of the uterine cycle is. It's the secretory phase, which is driven largely by progesterone. Okay. So that's what's going on in the uterus. I'm going to show you a picture in a little bit that'll highlight the uterine cycle in a little bit more of a time-dependent manner, and you'll see what I was talking about there. But just based on the micrograph, those are the layers of the uterus. Now I want to move on into the physiology that's running all of this. And what I just said, I'm going to show you the uterine cycle in over the 28-day cycle. And that's what this picture here is going to get at. So I'll show you that again a little later. But before I get to this stuff, which is about how the hormones regulate all this, I want to talk a little bit about hormones. Now, at this stage in the presentation, and I've always done this, I present a picture like this showing the chemical structure of a number of hormones. The point of this is not to ask you to memorize all these hormones or all these enzymes, but rather it's really the only graphical way I can represent what I want to talk about. Now, I'm trying to, when I don't have a picture from the book to demonstrate what I want to use, I'm trying to use copyright-free images. And so that's what this is. And the options are a little bit limited, so I have to use this one, which has way more information than I really want to talk about. If I could pare this down a little bit, I would. And I might on my own sometime. But we'll work with what we have here because it makes the point that I want. All of these are steroid hormones. And what defines something as being a steroid hormone is that it's derived from cholesterol, which is what we have up here. Cholesterol has these 3 6-sided rings and this 5-sided ring in this particular arrangement. And rings are really hard for us to make, so we usually get rings from molecules from our diet. But we modify cholesterol to use it. So there's an enzyme that cleaves the side chain off of cholesterol and gives us a different molecule. Okay. And then another enzyme changes this molecule into progesterone, which is the first thing that I actually want to point out here. There are other enzymes that will change it here and there into other things. Some of these are hormones that we've talked about in other systems, but we're just talking about reproductive function now. From progesterone, following a few enzymatic steps, we can make testosterone, which is what we have down here. Okay. In men, testosterone is sort of the main androgen hormone. It's made through several enzymatic steps from progesterone, and progesterone is a precursor to a number of other molecules also. Progesterone isn't really a hormone that's terribly active in the male reproductive system. It's just a precursor for testosterone. And we get down here. Now, I just said that testosterone is an androgen hormone. Another major androgen hormone is this one down below, which is called dihydrotestosterone, or DHT. Probably the only place you'd have run across any mention of DHT would be in a drug ad. Testosterone analogs like Propecia or Rogaine, or there are ads that are coming out more and more nowadays talking about low testosterone levels. So they might mention DHT, or dihydrotestosterone, which is just another androgen. And it's just a enzymatic step away from testosterone. Just one little thing's done different here. Also, one enzymatic step away from testosterone, we have estradiol, which is our main estrogen molecule that we've been talking about. The enzyme that's responsible for that, I was trying to zoom in on this, and the thing caused problems. But the enzyme that's responsible for that is called aromatase. An aromatic compound is a molecule, an organic molecule that contains a carbon ring with 3 double bonds in it, which is what we see on the estradiol molecule. And so what aromatase does is it takes testosterone and modifies it so that that one ring is an aromatic ring. It has all those double bonds. And in doing that, it has to change some of the other things about what's attached to testosterone there. So in testosterone, in that ring, we see just one double bond in here, and all the rest are single bonds. This carbon here, it's got 4 bonds like we'd expect for a carbon atom. 2 of them are within the ring, and 2 of them are double bonding to this oxygen. In that same position in the aromatic ring, that carbon has to have 3 bonds within the ring, and then 1 bond is left over to go to the oxygen. So the oxygen needs to bind to something, and it picks up a hydrogen, okay, which makes these 2 molecules chemically different. Okay. Estradiol is, chemically speaking, an alcohol, which is why it ends with OL on its name, and it has that structure right there. But and testosterone isn't that sort of thing. Exactly. Also up here, we have a carbon right where these 2 rings meet, and it's got 4 single bonds, each to other carbon atoms. And when the double bonds put in here, that carbon then is not going to be able to connect to this external carbon here, so that carbon's lost, and we get the double bonds in here. I've not really sat down to do a really fine counting of this, but I think we're talking about like 5 atoms different. I might be off by a couple of hydrogens, but that arrow that's pointing up next to where the cursor is right now, there's a carbon at the end of that, which has 3 hydrogens attached to it. So we take those 4 atoms off. And then there's some other hydrogens that are changed here and there in the ring, and I can't remember how many it is offhand. And then when we break the double bond to the oxygen, we add a hydrogen onto that oxygen. Okay, so I'm pretty sure that overall there's 5 molecules different between the 2. I mean, 5 atoms different between the 2 molecules. At at the chemical level, they're very, 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 very similar molecules. But at the physiological level, they're very, 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 very different results. Why? What does testosterone do? Oh, come on. What's testosterone do?
Too masculine.
Masculinizes the embryo, makes men. Okay. What does estradiol do?
Makes women nice and emasculated.
Feminizes, the appropriate way to say that. And actually, I don't know that estradiol is, in fact, the hormone that does that, but estrogens as a group would be feminizing. So very, very different results from very, very similar molecules. Why do they have such different results?
Function?
Well, that's what I'm saying. They have different functions. Why?
Is it the DNA, right? DNA.
No, they're steroids. They have nothing to do with DNA. So this is me at the end of the semester expecting you to remember something you learned earlier in the semester. So when early in the semester we'd be talking about something that had to do with these two molecules. Endocrine system.
Different hormone.
They are different hormones. They're very, very similar
Oh,
hormones.
wait.
but
Is
they
it
have-
one peptide, one
Nope, they're both steroids.
Can you ask the question again?
different company?
Why do testosterone and estradiol or estrogens in general have such different effects when they're such similar molecules? If I were to give you two molecules, one of them the glucose and one of them a modified form of glucose that's very similar, they both taste sweet. Why do these two hormones have such vastly different functions?
Different target.
What does it mean for a cell to be a target?
Estrogen receptor.
Yeah, different receptors. Testosterone binds to androgen receptors. Estradiol binds to estrogen receptors. And as a complex, that's where they're very chemically distinct and they have different effects. So, so there, yeah. Okay. Let me get the presentation back. I don't know what happened here. So what I want to do is, as soon as this comes back up, I want to go over and go back to looking at the ovarian cycle and the uterine cycle and think about how the hormones regulate those two together in lockstep. Okay. So first off, in this picture up at the top, this is kind of a representation of what's happening in the early follicular phase. Okay, it didn't fit in this space right here, so they put it all up here. But what it shows is actually a kind of an interesting thing to think about. The primordial follicles, there are a bunch of them. A woman is born with all of the cells that will ever be the egg cells that she produces. She's born with more cells than she needs. Okay? If I remember correctly, it's about 400,000 cells. And I can't remember if that's in one ovary or across both ovaries, but it's a lot of freaking cells. If you imagined the reproductive span in a woman's life going from, and these are just rough estimates to give us nice round numbers, from age 15 to age 50. Okay. And she's producing an egg every month, okay, for ovulation.
Yep.
So 35 times 12 is
420.
420.
Oh my God.
420 ovulatory events. 400,000 cells available to do that. Okay. Some people have the impression that menopause is when the ovaries dry up. No. There's plenty of cells left. Menopause is when the female system moves into a different stage, having to do with other aspects of reproduction, meaning really the survival of the species and raising those kids. If a woman's fertile all the way up to the end of her life, then she's going to have kids that have no mother. Okay, so part of the point of menopause is for survival of the species after birth, or survival of the offspring after birth. Okay. Anyway, so there's plenty of cells there. And actually, multiple cells will go into the the development of follicles. Only one will make it all the way through to the mature level at the tertiary follicle stage. That's what we see here. Okay. And then we have ovulation. And then and that's at day 14. At day 14, we split the ovarian cycle between the follicular phase and the luteal phase. And during the luteal phase, the corpus luteum secretes progesterone mainly, and eventually will degrade down to what's called the corpus albicans. Okay. I said corpus luteum describes the fact that it's yellow in color. Corpus albicans means that it's white in color, which means it's basically scar tissue. And if we look at the same thing for the uterine cycle, from day 0 to about day 5, the functional layer is detaches and leaves as menstruation, so that's menses. And then for the rest of the first half of the cycle, we're in the proliferative phase, which is when the functionalis grows thicker, which is mostly driven by estradiol. At ovulation, it's probably not quite to its full height, so it's going to keep growing a little bit, but more importantly, after ovulation with the presence of progesterone, that's going to stimulate the glands in the endothelium to start secreting what's needed for implantation and support of the early embryo. And then at the end, the tissue will start to slough off and detach, leading to menstruation in the next cycle. Okay. So those are the two cycles side by side. It'd be kind of nice if they'd sort of stretch this picture out a little wider so that 0 and 0 lined up. We have 14 and 14 lining up, and 28 and 28 lining up. Didn't, for whatever reason, do that. But, you know, plenty of room on either side to have done that. Wish they did. Now, down below, we look at the hormone levels, both those that are released from the pituitary and those that are released from the ovary. So the pituitary is releasing FSH and LH, and the ovary is releasing estrogen, meaning estradiol, and progesterone. At the beginning of the cycle, the pituitary hormones stimulated to be released by GnRH from the hypothalamus, their levels start to go up, and they're going to stimulate the development of the follicles. The follicles start to produce and release estrogen. As the estrogen levels start to climb, it acts as a negative feedback signal, limiting the release of FSH and LH. So those levels never get very high. But FSH is a little bit higher than LH because it's really the one that's stimulating the follicles. Then when the estrogen levels get really high, which is going to be around day 10 to 12, that peak of estrogen levels is actually going to switch from a negative feedback inhibition to the activation of eventually the release of all of the LH and FSH that had been stored in the pituitary gland as it was being inhibited. A lot more LH than FSH, so it's really responsible for causing ovulation. Okay. And then those levels fall off over the next several days. But it's called luteinizing hormone because after ovulation, then the corpus luteum is formed. Okay. Luteinizing hormone isn't causing the corpus luteum to do anything in here. It's just making it possible for it to exist after ovulation. Then when the corpus luteum is active, it's going to start releasing progesterone. It's also releasing estradiol, but not nearly as much as the follicles were before. Okay. So in the picture I was showing a little while ago with sort of the flow chart of the hormones, in the luteal phase picture, the third one, it just showed progesterone being released. But really, it's not just progesterone. There is estradiol, but fairly low levels of it. Okay. And those hormones will end up dropping off by the end of the 28-day cycle unless fertilization and implantation take place. And when they fall off, that's the signal to the endometrium to start detaching and sloughing off for menstruation at the beginning of the next cycle. Oh, why does it keep doing that? So that's a summary of putting those cycles together there and the hormones that play a role in it. This is back to the picture of the hormones I was using before. Like I was just saying, it says progesterone only in the luteal phase, but there's estradiol there. And then we see estradiol and the rest of it there. But that's what's sort of regulating things. It starts with the hypothalamus, which tells the anterior pituitary to release FSH and LH, which has an effect on the ovaries, which then release estradiol or progesterone to affect the endometrium of the uterus. Okay. If we go back to this picture here, which I showed you earlier, we see again that the hormones are the same for the male and the female system. And since this is a little bit simpler, there's not quite as many different phases going on or anything like that. They're showing us an anatomical representation of the hypothalamus and the pituitary, but still the same idea. So GnRH, not GnHR, released from the hypothalamus, stimulates the anterior pituitary to release FSH and LH. FSH stimulates sperm production in the tubules within the testes. And then LH stimulates interstitial or Leydig cells to release testosterone. What's not suggested here actually is testosterone is, its main function is really to help with the spermatogenesis effect. They just show its feedback mechanism here. But testosterone feeds back to the anterior pituitary and the hypothalamus, just like estradiol did in the previous phase, and for that matter, progesterone. So these hormones that are being released from the reproductive gland end up being a negative feedback signal. With estradiol, there is that time when high levels switch from being inhibitory to being an activator, but otherwise things are inhibiting. In this picture, they mention inhibin, which is another hormone that's being released from the reproductive glands. It's actually made at the ovary as well, but they left it out because it just sort of confuses the picture. But from the name, obviously, it's an inhibiting hormone. Okay, so it's just going to inhibit back to the pituitary and limit the release of those gonadotropin hormones. So that's happening in both systems. They only represent it in the male system picture here, but it's true. Okay. Okay, I actually got through what I wanted to, and I have a good half hour left. Wasn't expecting that. I really thought that it went longer than that. Question.
When they put like women on long-term birth control already, get it period for that long, what's happening?
Okay, actually, you remind me. Yeah, one of the things I skipped over trying to be fast was to talk about birth control pills. So let me go back to this picture here, because this is really where the birth control thing comes in. Birth control pills are a combination of progesterone and estradiol. And a lot of the more modern compositions have fairly low estradiol levels. because of data which is confusing, and I don't really quite know how to interpret it myself, but suggesting that estrogen levels can have some deleterious effects long term. So they're trying to limit the amount of estradiol there, but the progesterone is really the important part. So when you take birth control, the idea is you're taking these hormones to level so that your body thinks you're post-ovulatory. Okay, so you look like day 21. And so the ovaries don't produce any mature follicles, there's no ovulation, that's birth control. The traditional way that it's done is there's a week's worth of pills that do not have hormones in them. And not taking in any hormones, the levels go down like we see going to the day 28 point there. And that stimulates menstruation. Now, that traditional approach is based on this 28-day cycle, but it doesn't necessarily have to be done that way. So there's Seasonique, which is a regimen of taking the exact same pills, but you take the hormone-free pills only once every 3 months, okay, to cause menstruation just 4 times during the year, hence the name Seasonique, once for every season. Now, if you read the fine print or listen to the commercials carefully, they do say that it doesn't necessarily mean that there won't be monthly bleeding, because it is possible for spotting to occur because there's going to be a nice thick endometrium there. But it does limit the hormonal fluctuations and menstruation itself. So there are also some approaches that are even more long-term. I'm not familiar with the particulars of them timescale-wise, but I believe that there's a patch that might be yearly. Maybe I'm making that up. And I know that there's a type of IUD that has slow-release hormones in it. And IUDs are usually multi-year implantations, like 3 or 5 years, something like that. So that would suggest, and I really don't know too much about those, I'm more familiar with the traditional pill thing. But that would suggest that those are not going to have any fluctuations of hormone levels over the multi-year time that they're present. If there is such a thing as a yearly patch, which I might be imagining, and it's possible it could be yearly, but I'm pretty sure the IUD's 3 or 5 years or something. So that would suggest that you'd have a constant level of hormones there. I'm not positive what that really means for the endometrium, but if it was bad for the endometrium, it wouldn't be on the market. Ideally. But I'm really not that familiar with what's going on.
Wait, that's
versions. Yeah.
So if it's withholding for that amount of time, so say it's the year or 5 years, I don't know, like it's supposed to happen if you're stopping it from happening.
So if you look at the thing up at the top, proliferation is happening in the first half. If you're taking whatever form of birth control, that basically makes you stuck on day 21 hormone level wise, that puts you right in the middle of the secretory phase, which is not going to be endless growth of the endometrium. So it's not like it's going to keep growing and growing and growing. But it will keep secreting and secreting and secreting. And so I'm sure, just like I mentioned for Seasonique, any of those long-term hormone treatment birth controls probably have there some mention of the possibility of spotting, because there's going to be some fluid that comes from that. It's just not detaching the endometrium and then regrowing it. And from a purely tissue point of view, that can be helpful, because you got to expend energy to grow that tissue every month. So take that long-term hormone protocol might actually, in the long run, save a little bit of energy. And I don't know how much energy that is. It might be a few thousand kilocalories, a day's worth of food of energy. Like that. But, over the long term, it might have an effect.
You're like our personal guidance.
Personal data.
Yeah.
That's where I stopped.
So, no, this is all interesting because IUDs actually, I was looking for a non-hormone option. And IUDs, some of them are, but they put copper is released into your uterus and that prevents implantation. And how is that not bad?
It's 13 years.
How is that
13-year
not bad for
IUD.
you to put copper inside your?
Well, it's bad for you to put copper wire up there that you can see. It's a matter of, call it dosage or concentration. You have copper in your body. It's one of the trace elements that's in your body. It's toxic when it gets to be a high level. Any heavy metal is going to be toxic when it gets to a high level. So that's releasing a little bit of copper over the long term. What copper does is it binds to, or it plays a part in the structure of certain types of proteins. I'm actually not familiar with a particular example of copper, but I know zinc is an ion that's found in particular protein structures that are shaped to bind to DNA, and iron obviously is in hemoglobin. So metal ions do interact with proteins, and so it's releasing at a very low level that's still going to tape it at the trace element level, and it shouldn't be toxic. Again, ideally. It's not like you take a copper wire and use that as an IUD. That would be bad. But it's going to be a plastic IUD with copper in it that gets released slowly to interact with proteins, which I don't know what the proteins are, but they might interfere with follicle development, or I don't know. I'm making half this up, but it's just based on my understanding of physiology. Other questions? So I don't recall if I said this to you, because I did this stuff for my 11 o'clock class today also. Did I say anything about, when I was talking about X and Y chromosomes, did I say anything about there are a few other things that have to do with gender selection?
Oh, yeah.
I did say that? Okay. All right, so let me tell you a little story. A lot of these stories I have come from studies I've read about. Okay. So the story starts with the King of England. Okay. He was King Henry VIII of his name, for those of you who watch Game of Thrones. King Henry VIII. He married a very devout Catholic Spanish woman. Okay. And they did not have a male child, or actually they did, but it died very early on. So he did not produce a male heir. Okay. He was not happy about that. But they were married. She was a devout Catholic. She wouldn't let him get a divorce. He was King of England. He invented the Anglican Church, which is basically the Catholic Church plus divorce. It's not that simple. But anyways, so he had a bunch of mistresses / subsequent wives, depending on which religious orientation he felt himself to be at the time. There's a lovely, only minorly historically accurate series called The Tudors, which I think is on HBO or Showtime, where a very young, virile-looking Henry VIII is the main character rather than the fat slob, who's mostly the historical Henry VIII. But he was very upset because he was not producing, or his wives were not producing male heirs. Okay. He blamed them rather harshly in some cases. Sorry?
Must be all of them.
Yeah. Now, if you just look at the chromosomes, well, no, regardless of how you look at it, it's all his fault. But if you look at the chromosomes, it's about the X and Y chromosomes. Okay. The mothers can only contribute an X chromosome. So it's what type of chromosome is in the sperm that he's contributing. So it's up to him whether it's X or Y. The other thing that's his fault is because he's the king. Okay? There's a study that came out about 5 or 6 years ago now that sort of gets at this idea. They looked at women, and I think this was in a Scandinavian country or something like that, but they looked at women just across the population and asked them various questions, and the data that they considered for this publication was really just about what they had for breakfast. And so the women basically fell into two groups. Either they didn't eat breakfast or they did eat breakfast. And they followed these women and they collected data on how many children they had and what gender they were. What they found was women who did not eat breakfast were more likely to have male children. And women that did eat breakfast were more likely to have female children.
Wait, this is
Now, the numbers here are pretty minor. So it should be a 50-50 split. But the changes were like 55-45. So just a little bit more likely for women to have a male child if they didn't eat breakfast. And they also had another group in there. I don't remember the data that came from it exactly, but they looked specifically at women that ate sugary cereals for breakfast. And that went into some of their thinking. But the general conclusion that came out of this was about nutrition levels in the female system in terms of what's present in the uterine wall. Now, I was talking about the male reproductive system earlier, and I said that the seminal vesicle and the prostate gland secrete components of the seminal fluid, or the ejaculate, which includes in it actually fructose, not glucose, but as a carbohydrate monosaccharide to help power ATP production in the mitochondrion there. But once the ejaculate makes it into the female reproductive tract, the fructose present in the ejaculate diffuses away, and it's not really a good source for the swimming that the sperm need to do the rest of the way. So they rely on what they can find in the uterine lining. And the secretion that's taking place in the uterine lining includes, I'm not sure actually if it's fructose, but monosaccharides that can act as energy-rich molecules for the implanted zygote as it's undergoing some of its early cell division, that sort of thing. And the sperm, as they're swimming through, can make use of that too. Right? Now, when we're talking about a sperm and the chromosomes there, the difference in size between an X chromosome and a Y chromosome is actually pretty significant. The weight of a sperm cell carrying an X chromosome is significantly different than the weight of a sperm cell carrying a Y chromosome, just a lot less DNA in it. And so it takes more energy for an X-bearing sperm to swim. It's got to move more mass. So back to Henry VIII, assuming that he wasn't completely cruel, I'm sure he let the women in his life eat nice, healthy breakfasts every morning. And so they had very well, I'm talking myself in the corner. They had great nutrition. So the secretions in the uterus are going to be very rich in nutrient molecules. And those molecules will help to power his sperm to swim along. That's going to cut down on the likelihood of his Y chromosome carrying sperm to make it all the way to the egg cell. Again, it's a minor difference, but 55-45 split there, but it can play a role in that. Yeah.
Wait, the Y is heavier, so it had
The X is heavier.
Okay.
So it needs more energy to swim. Okay. So he feeds the woman very well. Therefore, his heavier X chromosome carrying sperm can swim as well as a Y carrying sperm. That's sort of where they're going with that study. As opposed to a woman who does not eat breakfast and therefore she's not going to secrete as nutrient rich of secretions in the uterus, and therefore Y chromosome carrying sperm can swim farther because they have less mass on the limited resources they have getting through the uterine system. That's the basis there, yeah.
And so wouldn't they take that out and then do the whole third world crap that they always do and say that it'd be all like the boys in like a poor country and all girls in a rich country?
Well, the data is only coming from like I said, I think Scandinavia. So they didn't have any data looking at other situations, and they're doing a lot of extrapolation from the data as it is. But then carrying it over there is a bit much. And also, you have to consider that there are some cultural issues. Female children in China can be abandoned, and so it'll look like there's more male children there, et cetera, et cetera. So yeah, they didn't quite push the data that far, but they pushed it pretty far as it was. Now, without getting into the modern cultural aspects of things, they did talk about the reasoning behind this probably, which is reproductive strategy is how well you can spread your genes throughout the human population. If you're a woman and you're going to have a kid, you are more successful spreading your genetic material through the population in different ways depending on the availability of resources in the environment. Okay. If resources are good, it's best to have a daughter who you can put a lot of energy into having her survive, and she can put a lot of energy into having children. And therefore, all of your grandchildren are most definitely yours. So it's a great way to make sure your genes make it into the population. When there's sufficient resources for the gestation of a female child and her subsequent ability to survive and have children of her own. If resources are low, you have to invest energy in gestating your child. But if resources are low, having a male child is better because then he can, at very little energy expenditure, spread your genome far and wide. Okay. The trade-off is that you can't be positive that your genes make it out into the population. There's no definitive way, short of testing DNA now, to be able to know that your grandchildren from your son are actually yours. Okay? If your son was cuckolded and your grandchildren are actually somebody else's kid who he's raising as a child, you might not be passing on your gene. But if he's prolific, then you're going to get a lot of genetic information out there, even though resources are limited. That's kind of the idea. Again, it's a fairly minor difference because if resources are low environmental wide and everybody started having kids, started having male kids, then there would be no subsequent generation because you need 2 to tango. Okay? So somebody's got to be having female children. Okay? So it's a minor change. Again, normally it should be about 50-50, but it ends up being about 55-45 split, one way or the other. So that's the idea. And so back to Henry VIII, he, being the king, is living in a resource-rich environment, as are his wives, concubines, mistresses, whatever. And so they're more likely, by a little bit, to have female children based on this data. Not at all really explaining one way or the other, but it does put a lot more blame on him for cutting their heads off. Okay? That it's his sperm and his ability to provide that's most likely contributing to the higher likelihood of him having daughters.
So what should have done? Stop them.
Sorry?
What should have done? Stop.
Yeah, if you're really going to overextend the data, that would be the most obvious thing, but I would not recommend that. He just shouldn't have cut people's heads off.
Yeah.
Yeah, like even before that.
Yeah, generally that's a good rule to live by, but yeah, he didn't quite.
It's the light, yeah.
He didn't have the moral justification he thought he had before that. I mean, he's the king, so morals are kind of his thing. He's the.
Apparently not.
No, not that in an absolute sense, but being the king, being divinely chosen to be the leader of England, what he says is kind of the moral code. Isn't exactly true. But. So that kind of gets at some suggestions that it's more than just the XY chromosome determination. There are other things that play a role. Yeah.
What about people that are born with
I don't really like to get into that stuff too often because it gets kind of weird. Not in a don't do a Google search kind of weird, but just it's kind of hard to really explain it exactly. But what's happening is if you go back to meiosis, it should be in the second meiotic division that the duplicated chromosomes split and one goes into each gamete. Actually, it doesn't have to be in the second meiotic division. Somewhere in the division, it should be everything splitting equally. But sometimes something splits unequally. And so, yeah, some stuff sticks together and you get 2 copies of chromosome in a gamete when you should have only had 1. And when that happens with the X or Y chromosomes, then there's something that happens. Now, the X chromosome is pretty large, and so multiple X chromosomes are unlikely to be a regular condition. I'm not entirely sure, but the most common condition that's multiple sex chromosomes is XXY, which is the egg cell has an X, it's always going to have an X, and the sperm has an X, but the Y sticks around too. So sperm's actually delivering both. Having a Y chromosome causes masculinization of the embryo, but having 2 Y chromosomes also causes some feminization things to happen, and so that's.
So the child would more likely be female, physically, like features wise, but anatomy would be male.
Actually,
Or the other way around.
mostly will look female. Okay. Likely to be infertile, because things are just so messed up that work. Now, it's been a long time since I really looked at this, but if I remember correctly, XXY is the supermodel genome. Okay. There's a high incidence of supermodels having XXY chromosomes, if I remember
Was that
this correctly.
episode House?
Because anatomically, it's feminized, but some of the masculinization actually causes some
So
body
the height
structural
would come
changes,
from-
height, and that kind of thing. Again, if I remember correctly, I might be misremembering exactly what it is, but I'm pretty sure it's XXY that's the supermodel condition. So, but I don't believe that they're fertile in comparison. But I'm not sure about that. It might be possible.
How do twins get produced? Like, twins?
So there are different possibilities. Fraternal twins are when there are 2 eggs and 2 sperms. Identical twins is when there's 1 egg and 2 sperms. So if the egg in its initial division, or wait, no, I said, sorry, it's not 1 egg, 2 egg. When a fertilized egg divides and gets kind of overzealous in its division and separates, those 2 resulting cells can each give rise to a separate embryo. It's when they're actually connected that they stay part of a single embryo. But if they divide and they physically separate from each other, they'll each become their own embryo. And they're clonal copies of each other. So they're identical. Fraternal is when 2 ovulations take place, and so 2 eggs are fertilized, and you get 2 offspring who are really just brother and sister or brother and brother, genetically speaking. They're not at all identical, but they have the same birthday. So that's about as much as I know about weird sex chromosomes and sorts of things. But if you're interested, feel free to do some searches. Just be careful on Google what you search for when you're talking about sex chromosomes. So that wraps things up. Remember, next week on Thursday is technically a Wednesday schedule, so we do not have class. And then the Thursday after that is the final exam period for this class. You'll be coming in at 1:30 for that. Okay. So otherwise, have a nice couple of weeks of getting everything done.